Hello, welcome to Biology, the scientific study of life. Linked genes tend to be inherited together because they are located near each other on the same chromosome. The number of genes in a cell is far greater than the number of chromosomes. In fact, each chromosome has hundreds of thousands of genes located on the same chromosome that tend to be inherited together in genetic courses are said to be linked genes. When geneticists follow linked genes in breeding experiments, the results deviate from those expected from Mendel's law of independent assortment. How linkage affects inheritance, scientific inquiry. To see how linkage between genes affects the inheritance of two different characters, let's examine another of Morgan's Drosophila experiments. In this case, the character are body color and wing size, each with two different phenotypes. Wild-type flies have gray bodies and normal-sized wings. In addition to these flies, Morgan had double mutant flies with black bodies and vestigial wings, much smaller than normal wings. The alleles for these traits are represented by the following symbols: B positive, which mean gray, B which mean black, VG positive, which means normal wings, VG which means vestigial wings. The mutant alleles are recessive to the wild-type alleles, and neither gene is one Essex chromosome. In studying these two genes, Morgan carried out the courses shown this figure in the next page. He first mated tree breeding wide type flies B positive, VG positive with a black vestigial winged ones BBB, VG to produce heterozygous F1 dihybrids, then coursed female dihybrids with the two breeding males of the double mutant phenotype BBB-VG. In this second course, which corresponds to Mendelian test course, we know the genotype of the female parent B positive BB, VG positive VG, and we also know which allele combinations are paternal, meaning derived from the parents to the in the P generation B positive with the VG positive and B with VG. We don't know, however, whether the two genes are located on the same or different chromosomes. In the testicles, all the sperm will donate recessive allele B and VG, so the phenotypes of the offspring will depend on the ova's alleles. Therefore, from the phenotypes of the offspring, we can determine whether or not the parental allele combinations B positive with the VG positive and B with the VG stay together during formation of the F1 female's ova. When Morgan's cohort classified according to the phenotype 2,300 offspring from the test-cross mating, he observed a much higher proportion of paternal phenotypes than would be expected if the two genes assorted independently. Notice also this as it's shown here in this figure. Notice also, based on these results, Morgan reasoned that body color and wing size are usually inherited together in specific combinations, the paternal combinations, because the genes for these characters are on the same chromosome. However, if the genes for body color and wing size were always inherited together in those paternal combinations, no non-paternal phenotypes would have been observed among the offspring of Morgan's test-cross. In fact, both of the non-paternal phenotypes were produced in Morgan's experiments, suggesting that in this figure, notice that suggesting that the body color and wing size genes are only partially linked genetically. To understand this result, we need to further explore genetic recombination, the production of offspring with combinations of traits differing from those found in either parent, genetic recombination and linkage. As we have learned, the meiosis and random fertilization generate genetic variation among offspring of sexually reproducing organisms. Here we will examine the chromosomal basis of recombination in relation to the genetic finding of the Mendel and Morgan. Recombination of unlinked genes, independent assortment of chromosomes. Mendel learned from courses in which he followed two characters that some offspring have combinations of traits that do not match those of either parent. For example, we can represent the cross between a plant with yellow round seeds that is heterozygous for both seeds color and seed shape. Notice that WR capital and WR small and so on. And a plant with the green wrinkled seeds homozygous for both recessive alleles Y, notice that Y small, R small, by the following Punnett square. Notice the gametes from yellow round heterozygous parent Y capital small. Notice that in this Punnett square that one half of the offspring are expected to inherit a phenotype that matches one of the parental phenotypes. These offspring are called paternal types, but two non-paternal types are also found among the offspring because these offspring have new combinations of seed shape and color. They are called recombinant types, or recombinants for short. When 50% of all offspring are recombinants, as in this example, geneticists say that there is a 50% frequency of recombination. The predicted phenotypic ratios among the offspring are similar to what Mendel actually found in Y capital Y small, R capital R small by Y small, R small courses. A 50% frequency of recombination is observed of any two genes that are located on different chromosomes. The physical basis of recombinations between unlinked genes is the random orientation of homologous chromosomes at metaphase 1 of meiosis, which leads to the independent assortment of alleles, as is shown here in this inquiry. A combination of linked genes crossing over. Now let's return to Morgan's fly room to see how we can explain the results of the Drosophila testacross illustrated in this figure. Recall that most of the offspring from the testacross for body color and wing size had parental phenotypes, suggesting that the two genes are on the same chromosome. But a small number of offspring were recombinant. Although there was linkage, it appeared to be incomplete. Faced with these results, Morgan proposed that some process must occasionally break the physical connection between genes on the same chromosome. Subsequent experiments demonstrated that this process, now called crossing over, accounts for the combination of linked genes in crossing over, which occurs while replicated homologous chromosomes are paired during a prophase of meiosis 1. One maternal and one paternal chromatid break at corresponding points and then are rejoined to each other. Notice this one. Boom. In fact, the end portions of two non-sister chromatids trade places each time a crossover occurs. The recombinant chromosomes resulting from crossing over may bring alleles together in new combinations, and the subsequent events of meiosis distribute the recombination chromosomes to gametes. Shows how this figure, notice that, shows how crossing over in dihybrid female fly resulted in recombinant ova and ultimately recombinant offspring in Morgan's testicles. Most of the ova had a chromosome with either B positive, VG positive, or B and VG paternal genotype for body color and wing size, but some ova had a recombinant chromosome B positive, VG, or VG positive. Fertilization of these various classes of ova by homozygous recessive sperm BVG produced an offspring population in which 17% exhibited non-parental recombinant phenotypes. As we discuss next, the percentage of recombinant offspring, the recombination frequency is related to the distance between linkage genes. Back to the inquiry, are the genes for body color and wing size in fruit flies located on the same chromosome or different chromosome? Experiment. Morgan first mated two breeding wide type flies with black. Notice that B generation homozygous, B positive, VG positive, notice also testicles. The result B, notice also the result, a paternal type of spring recombinant, non-parental type of spring, notice also black normal, gray, notice the number also. Okay, the conclusion also, and these two genes were on different chromosomes, the alleles from the F1 dihybrid would sort into gametes independently, and we would expect to see equal numbers of the four types of offspring. If these two genes were the same chromosome, we would expect each allele combination B positive, VG positive, and B and VG to stay together as gametes formed. In this case, also only offspring with paternal phenotype would be produced, so most offspring had a paternal phenotype. Morgan concluded that the genes for body color and wing size are located non-paternal phenotypes indicate located on the same chromosome. However, the production of a small number of offspring with non-paternal phenotypes indicated that some mechanism occasionally breaks the linkage between genes on the same chromosome. Okay, thanks for